Good morning, good afternoon, everyone. In this presentation, I'm going to give some findings of my PhD research work, which was about anti-cancer activity of the Graviola fruit extract against different type of cancer cell lines, breast MCF-7 and colon HT-29, using in vitro and in vivo methods. This research work was under Biotechnology Engineering Department, Faculty of Engineering, IIUM Malaysia. So this is the outline of this presentation, starting with introduction, literature review, research methodology, and the result and discussion. At the end, we have conclusions and recommendations. So what is Annona mericata? Scientifically named Annona mericata, which is known as Graviola, Soursop, Guanabana, depends on the different regions around the world. This Graviola is a tropical evergreen fruit belonging to the Annonaceae family. It's grown and distributed in tropical and subtropical regions around the world. This fruit, as a ripe fruit, is eaten as raw or processed into ice cream, jelly, nectar, and fruit juices. For medicinal use, this fruit is used to treat fever, flu, inflammations, diarrhea, and different bacterial and parasitic infections, and also to treat hypertension and cancer disease. So this fruit, the fruit, as mentioned in this figure, is large, heart-shaped, and green in color. More than 200 phytochemicals were extracted and isolated from this fruit, especially alkaloids, phenols, and terpenoids, flavonoids, tannins, and the most important one is acetogenins. This fruit also has a long history of traditional use around the world. So the problem statement of this research, as we know that cancer is still causing or the second cause of the mortality in the world, with an estimation to rise by at least 70% by 2030. And also we have the conventional treatments of this disease rely on chemotherapy, radiotherapy, surgery that cause various kinds of undesirable side effects. And these chemotherapeutic drugs are very expensive and not affordable for everyone. On another hand, alternative treatments to develop a new alternative anti-cancer drugs from plants is remaining one of the most challenging area of research. And for Annona mericata, as we know that this fruit has various pharmacological properties, which is not discovered yet, and most studies were reported on the leaves and seeds and barks and stem of the tree, while less focused on the anti-cancer property of the fruit itself. And for the extraction methods, graviola were extracted by different extraction methods, such as soxalate and maceration, that are not safe for human consumption because they are used organic solvents. So maybe ionic liquid microwave assisted extraction may be a better choice in producing safe compounds for human consumption. So the objectives of this research, starting with the main objective, which is to determine the antiproliferative effect of the crude extract and the fractions of the graviola fruit on in vitro breast MCF-7 and colon cancer cell lines, which is HT-29, and in vivo zebrafish developing embryos by fulfilling the following objectives. We have 5 objectives, starting with the first, which is to determine the optimized processing conditions of the ionic liquid microwave assisted extraction for yield to enhance the percentage yield and cytotoxicity effect using the design expert software. Second is to fractionate and semi-purify the crude extract of the graviola fruit using thin layer chromatography and column chromatography to enhance the cytotoxicity. The third objective is to investigate the in vitro and in vivo behavior of a human breast MCF-7 and colon HT-29 cancer cell lines and normal VERO healthy cells and in vivo zebrafish developing embryos in response to crude IL-GFE, which is the ionic liquid graviola fruit extract treatment through the antiproliferative assay and acute toxicity assay. And the fourth objective is to identify the crude IL-GFE mechanism of action in inhibiting MCF-7 and HT-29 cancer cell proliferation via flow cytometry technique. And at the end, the fifth objective is to carry out an in vitro investigation on the effect of the crude IL-GFE metabolomic profile of human cancer cell lines using GC-TOFMS-based metabolic profiling and chemometric methods. So for the literature review, as we know that cancer is among the most common cause of mortality in the world, while however conventional treatments cause many undesirable side effects such as hair loss, hormonal fluctuation, depression, and even death. So on another hand, graviola possesses a wide range of biological activities. But there are limited studies on graviola fruit. As I said before, that different studies were focused on the leaves and the barks and seeds of the tree, and less focused on the fruit. So for the graviola also exhibits a great anticancerous properties, but less focused on the anticancerous property of the fruit pulp of graviola as reported on cancer cell lines. And we have from literature more than 120 anonaceous acetogenin have been isolated from leaves, bark, seeds, and roots, but little attention has been paid to the anti-cancer property of acetogenin contained in the edible fruits. And previous study have reported the mechanism of inhibition of the acetogenin as the main and the major compounds in the Annonaceae family. However, the mechanism of the whole fruit inhibition toward cancer cells is still lacking, and most graviola fruit extractions were performed through conventional extraction methods. There are no phytochemicals extracted by ionic liquid microwave assisted extraction as a new method and new approach to extract safe biocompounds from plants. So also, as to our knowledge, there is no study conducted the kinetic behavior of the cancer cells in response to graviola fruit extract. Also, no reported study has assessed the possible toxicity of the graviola fruit extract on in vivo zebrafish model, and also there is no metabolomic study investigated the metabolic profiling of the cancer cells in response to graviola fruit extract treatments. So these tables present and summarize different findings from literature. And this the flow diagram of the major experiments used in this research, starting with the extraction of the biocompounds from graviola fruit using ionic liquid. And secondly is optimizing the extraction parameters of the ionic liquids and the microwave assisted extraction. Then after that, we have semi-purified and fractionated the crude extract of the graviola fruits using different solvents. At the end, we have done the phytochemical screening using different tests in order to screen for the acetogenins, alkaloids, terpenes, and phenols and flavonoids. After that, for the third objective, we have used different studies. For the in vitro study, we have used cell culture of the MCF-7 and HT-29 cells and VERO cell lines to evaluate the antiproliferative of the crude extract graviola fruit and its different fractions. After that, we have investigated the growth kinetic of the cancer cells in response to the crude extract. On another part, the in vivo study have used the zebrafish developing embryos in order to assess the toxicity of the crude extracts on in vivo model. For the first objective, we have used the flow cytometry technique in order to assess for the cell cycle phase distribution and apoptosis and necrosis induction in the MCF-7 and HT-29 cancer cells in response to the graviola fruit extracts. For the fifth objective, we have used the GC-TOFMS metabolomic profile in order to assess the alteration of the different metabolites in response to the crude extracts using XL state software. So this is the overall methodology that were used in this research. We start with the first objective, which is to determine the optimizing processing conditions of the ionic liquid microwave assisted extraction. We have used firstly, we have prepared and collected the fruits of the graviola from Anticancerous farming from Selangor, cleaning and freeze drying the fruits, then extract the biocompounds using ionic liquid microwave assisted extraction, then optimize the extraction parameters of extraction time and irradiation power and solid liquid ratios in order to enhance the responses, which are percentage yield and IC50 on microgram per milliliter, using different formulas. And this table summarizes the different parameters or independent variables and different levels of the independent variables in order to enhance the two responses, which are yield and IC50. So the result of this of the optimization we have in this table the experimental design obtained from the FCCCD, consistent of the 12 replicates of the experiments in order to enhance the percentage yield and IC50 of IL-GFE on MCF-7 cells and the IC50 of IL-GFE on HT-29 cells, which is the colon cancer. We have here the different parameters with the experimental design, and for the percentage yield, we have got that the optimum extraction conditions of the time and irradiation power and solid liquid ratio have given the 66% of the yield, and for the IC50 of the IL-GFE on MCF-7 have given an IC50 of 8.12 microgram per milliliter toward MCF-7 and 11.85 microgram per milliliter toward HT-29. So for the statistical analysis of the experimental results, we have the ANOVA for response surface model with the extraction yield. We have that the model was significant and the lack of it was not significant, and also the coefficient of determination was near to 1 and near to the adjusted R-square. And this is the 3D response surface plot. We have different interaction between time and power, time and solid liquid ratio, and power and solid liquid ratios. We have in this figure B that the interaction between time and solid liquid ratio was given a high extraction yield. And for validation, we have used the optimum extraction parameters that were predicted by the model to validate this model, and have given 66% of the graviola fruit extract yield. So the same optimization was done in order to enhance the IC50 of the crude ionic liquid graviola fruit extracts on MCF-7 and HT-29, and the models were significant, and R-square also was quite high, which which is given the significance of the model and the results. To validate this, the IC50 results, we have here in this table the optimum extraction conditions in order to give the best results of the IC50 on MCF-7 and HT-29. We have validated this optimum extraction conditions and given at the end the result of IC50 of 4.75 microgram per milliliter against MCF-7 and 10.56 microgram per milliliter on HT-29 cancer cell lines. And these results were concluded from these figures. In this figure, we have the percentage of MCF-7 cell viability versus concentrations of the Graviola fruit extract, and we have concluded the IC50 from the formula, which was the 4.75 microgram per milliliter. Same with the graph of the HT-29 cancer cell viability versus concentrations of the IL-GFE, which has given the IC50 of 10.56 microgram per milliliter. On another hand, we have also assessed the toxicity of the IL-GFE on VERO cell viability, which is normal healthy cells. We can see here that the IL-GFE doesn't give an IC50 for these cells, which means that the crude IL-GFE showed selective antiproliferation against cancer cell lines, while it was safe toward normal healthy VERO cell lines, as you see here. For the second objective, we have separated and semi-purified the crude extracts using different methods, which are thin layer chromatography, in order to determine the best solvent type and the ratio for separation of the sample. And the second method, which was column chromatography, as is observed here in this figure, in order to fractionate and separate the different fractions from the crude extract. And the result of this purification is summarized in these tables. For the first of the table, we have different solvents, which are hexane, dichloromethane, methanol, and water, with different ratios, have given different fractionation. And based on the TLC-RF values, we have combined them based on their RF values. At the end, we have got 6 fractions, from fraction A until fraction F. In this table, we have the IC50 of the IL-GFE and different fractions on MCF-7 and HT-29 cell lines. This table summarizes the IC50 of different fractions and the crude extracts toward MCF-7 and HT-29. We can see here that the best IC50 for MCF-7 was given by the crude extracts, IL-GFE, while the different fractions have given more IC50 than the crude extracts. As we know that the best IC50 is the minimal one. The minimal value presents the best IC50. And also for HT-29, we have here the crude extract has given 10.56 microgram per milliliter, while other fractions have given more values than the value that given by the IL-GFE. We can conclude that the crude IL-GFE showed better performance in reducing the IC50 of both cancer cell lines compared with the 6 purified fractions. Therefore, the crude extract was used for the whole experimental of this study. We have continued the other experiments with the crude extract. For the third objective, we have used to investigate the in vitro behavior of the human breast MCF-7 and the colon HT-29 cancer cell lines in response to the crude treatment of IL-GFE using cytokinetic study. In this curve, we have different type of treatment. We have the control of, or the untreated MCF-7 in blue line. We have the MCF-7 treated with the crude extract IL-GFE in red color. And we have the MCF-7 treated with Taxol as a positive control. We know that Taxol is the chemotherapeutic drug commercially used, which is given here with a green color. So from this curve, we have calculated the number of generations X and specific growth rate mu and doubling time. We can see here that the number of generations was decreasing after treatment with the crude IL-GFE and Taxol, while the doubling time was increasing from the control until the crude IL-GFE treatments. So this representative photograph of the untreated MCF-7 cells and the treated MCF-7 cells with crude IL-GFE in a time-dependent manner. So we can see here that the density of the cells were decreased after treatment with the crude IL-GFE in a time-dependent manner, and the dead cells was increased, especially after 120 hours. We can see here that the dead cells was increased compared with the control at the same time. This is for MCF-7. Same result with HT-29, which is colon cancer cell lines. This is the curve of the untreated HT-29 and the treated one with crude IL-GFE and the treated with Taxol. In this table we have the number of generations X and the specific growth rate, which was also decreasing, and doubling time was increasing after treatment with the crude IL-GFE and Taxol. And with these formulas, we can calculate the volume of the cancer cells at any time for the untreated HT-29, and for the treated HT-29 with the crude IL-GFE, we can calculate the inhibition of these treatments, and we can calculate also the estimation of the treatment's effectiveness with this formula after treatments and before treatments. This also representative photograph of the untreated HT-29 in this part, and the treated HT-29 with crude IL-GFE. Same conclusions, that we can see here that the density was decreased after treatment with the crude IL-GFE, and the number of death cells were increased. We can conclude that the crude IL-GFE had inhibited the growth kinetic of MCF-7 and HT-29 by reducing the number of cell generations and increased the doubling time of the cells, or of the cancer cells. Also, for the third objective, for the in vivo study, we have used, as I said before, the zebrafish model in order to assess the toxicity of the crude IL-GFE using acute toxicity study. We have used different endpoints in order to assess this toxicity, such as death and coagulation, heartbeat rates, edema, body curvature, detachment of dye, lack of somate formation, and hatching time. Using the typical 96 well plate configurations, we have in these photographs the embryo at the 24 hours post-fertilization HPF, and a larvae at 72 hours post-fertilization, and adult zebrafish. We have used in our study the embryo from 0 HPF until a larvae at 120 HPF. So the result of this study, we have here the survival rate of the zebrafish developing embryos versus the concentrations of the IL-GFE crude extracts. We can conclude here the LC50, which is the lethal concentrations, 50% of the lethality concentrations. We can calculate it from this formula, and the survival rates versus the time with different concentrations of the crude ionic liquid Graviola fruit extract. And in this graph we have the hatching rate in a time-dependent manner. So we can conclude from these two figures that the crude IL-GFE exposure had reduced the survival rate of zebrafish larvae with an LC50 of 125 microgram per milliliter. And the hatching rates of 48 hours post-fertilization average was in line with the standard hatchability of the zebrafish model. And for in this figure we have the number of heartbeat rates versus concentrations of the IL-GFE. We can conclude here that the heartbeat rates were recorded at, of course, at 96 HPF, and showed inhibition of the heartbeat rates by 35% at 125 microgram per milliliter of the IL-GFE compared with the control group. So we have 35% inhibition of the heartbeat compared with the control. This is increasing concentrations of the IL-GFE on microgram per milliliter. For the fourth objective, we have used the flow cytometry technique to identify the crude IL-GFE mechanism of action in inhibiting the MCF-7 and HT-29 cancer cell lines. So we have used different staining methods. We have used the RNA-PI staining assay in order to assess the induced cell cycle phases distribution, and we have used Annexium-5 PI staining assay in order to assess the apoptosis and necrosis induction in MCF-7 and HT-29 cancer cell lines using the flow cytometry technique. So for the result of the RNA-PI staining assay on MCF-7 cell cycle distribution, we have here the cell cycle distribution in a time-dependent manner. This is for the control, and this is after 24 hours treatment, and this for after 48 hours treatment, and this is after 72 hours treatment with crude IL-GFE. So in this plot we have the different phases, which is presented here as sub G0, and this presenting the G0 G1 phase, this presenting the S phase or synthase phase, and this presenting the G2M phase with different percentage in a time-dependent manner. In this result were summarized in this bar chart. We have the percentage of cells at each phase and different phases with control and different time of treatments. We can see here that the percentage of cells at sub G0 were increased from the control until 72 hours treatment. Same with G0 G1 phase. You can see here that the number or percentage of cells at the G0 G1 phase were increasing from the control until 24 hours treatment, 48 hours and 72 hours treatments were increasing, while for the S phase and G2M were decreasing from the control until 72 hours treatments, same with G2M. So this for MCF-7, and this is same for the HT-29 is summarized in this figure. The sub G0 phase was increasing from the control until 72 hours treatment, while also for the G0 G1 was increasing, while for the S phase and G2M were decreasing from the control until the 72 hours treatment. This is the result of the cell cycle assay. For the apoptosis and necrosis assay, we have here the percentage of apoptotic MCF-7 cells treated with crude IL-GFE at the corresponding IC50 for 24 hours treatment, 48 hours treatment, and 72 hours treatment, and this is the control. In this plot, we have the live cells in this part, we have the necrotic cells in this part, we have the apoptotic cells in this part. So we can see here that the live cells was decreasing from the control until 24 hours treatment, then 48 hours treatment, then 72 hours treatment. The percentage for the control was 93% of living cells, decreased until 16% of the living cells. On another hand, for the necrotic cells, the percentage of necrotic cells for the control was 5% and increased after 24 hours treatment until 15%, then until 68% after 72 hours treatment. And for apoptotic cells, we have here for the control 0.32% apoptotic cells in the control, or the untreated cells. After treatment for 24 hours, increased a little bit, then increased to 5% after 48 hours per treatment, then to 14% after 72 hours treatment. We can conclude that the crude IL-GFE arrested the MCF-7 and HT-29 cell cycle at the growth static G1 phase, which explains the antiproliferation of cancer cells by the treatment. And the Annexin 5-PI staining assay showed that the IL-GFE has remarkably induced apoptosis and necrosis in breast cancer cell lines. Finally, for the fifth objective, we have investigated the effect of the crude IL-GFE on the metabolomic profile of the human cancer cell lines using the GC-TOFMS-based metabolic profile and chemometric methods. We have summarized the methodology here, starting with cell culture, MCF-7 and HT-29 cell culture, then quenched after 72 hours treatment at the corresponding IC50. We have quenched the cell metabolism, then extracted the metabolite from the cells, then analyzed them using the GC-TOFMS spectrometry. Then the result and the data of the MS spectrometry have been analyzed using Excel state software to do multivariate data analysis. So the results of the multivariate data analysis, starting with the agglomerative hierarchical clustering dendrogram, which has grouped at least three clusters with a great dissimilarity of about 450. This is for MCF-7 and this is for HT-29. In order to this also, we have used the first and second principal component analysis to summarize this result. We have at least 4 groups of metabolites have clustered in a scatter plot, accounted for 77% for MCF-7 and 85% for HT-29. We have clearly 4 groups of metabolites. So for the result of the plot of loading formed by the first 2 principal component analysis, we have here in this scatter plot different metabolites that presents a synergistic effect toward other metabolites, and metabolites that has the antagonistic effect toward other metabolites. We have the model. In this model, the statistical model, Kaiser-Meyer-Orkin test for sampling adequacy was presenting 0.52, and the Bartlett's test of sphericity, which was highly significant, which indicate the suitability of the obtained data for factor analysis. PCA illustrates the correlated biomarkers with each group of metabolites. We have 14 out of 22 metabolites have showed a strong correlation among other metabolites with a factor loading between 0.7 and 1. The metabolites that have a synergistic effect are presented in this part, such as alanine, aminoguanidine, benzoquinolene, and propanoic acid, while the antagonistic effects metabolites were carbamic acid methyl ester and carbohydrazide and etc. This is for MCF-7. Also for HT-29, this is the plot loading formed by first two principal component analyses. We have the KMO test of 0.52 and Bartlett's test of sphericity, which was significant. And the PCA have summarized 43 out of 55 metabolites showing strong correlation among other metabolites. And this is the different metabolites that have synergistic effects and the metabolites that have given antagonistic effect among other metabolites. For the pathway analysis of metabolomic profile, we have concluded that many different metabolites or metabolic pathways were altered after profiling data analysis. We have here, for example, ketone bodies metabolism were altered since the trifluoromethyl-based methyl ketone was downregulated, and also the steroid hormone biosynthesis were altered since the pregnenolone one has downregulated. We have also the biosynthesis of amino acids were altered since the acetic acid aminocarbonyl was downregulated after treatment with crude extract IL-GFE. We have also the pentose phosphate pathway was altered since the altonic acid lactone was upregulated, and the central carbon metabolism was also altered since the propanoic acid was downregulated. All of these metabolic pathways are integrated in the energy production, growth of the cancer cells, and the supply of biosynthetic precursor and energy. So as we can see here that the healthy cells normally catabolize glucose in the presence of the oxygen to pyruvate. Then the pyruvates enter to 3-carboxylic acid cycle in order to produce 36 ATP. While the tumor cells, even in the presence of the oxygen, glucose catabolized to pyruvate, then ferment to lactate. This pathway gives only for ATP, which is known as Warburg's effects. That's why the cancer cells use the different metabolic pathways in order to produce more energy, such as fatty acid and amino acid metabolism. In conclusion, the pathway analysis of the metabolomic profile showed an alteration of many metabolic pathways that contribute to energy metabolism and the growth of cancer cells. So the previous result was summarized and concluded from the KEGG online database, and we have also used the Ingenuity Pathway Analysis in order to conclude the altered pathways after treatment of the MCF-7 by the crude IL-GFE, and the altered metabolites and metabolic pathways are summarized in this schematic overview. We have here different metabolites in different color. We have a green color presents the downregulated metabolites, and the red color presents the upregulated metabolites. green color present the non-detected metabolites, and we have the predicted inhibition and the predicted activation by the software, Ingenuity Pathway software. We have here that the glycolysis pathway was altered since the pyruvate was predicted to be inhibited, and this pyruvate has inhibited the acetyl-CoA biosynthesis, which is responsible for the entering in the citrate cycle and producing the ATP. Also, the pyruvate fermentation to lactate has altered because the lactate was downregulated, which means that the both pathways, glycolysis to citrate cycle and the fermentation, was altered after treatment of the MCF-7 with crude extract. Also, different amino acids such as alanine, aspartate, and glutamine metabolism, glycine, serine, and threonine metabolism, threonine degradation, glycine biosynthesis, all of these amino acid metabolism were altered since different metabolites were altered and downregulated after treatments. We have also the steroid hormone biosynthesis also was altered since the cholesterol was downregulated, which is the precursor of the pregnenolone that is included in the steroid hormone biosynthesis. And also the oleate biosynthesis was upregulated. The oleate was upregulated, so oleate biosynthesis was activated and in this after treatment. Also for the glycine and 6-phenyl-CoA was downregulated, which means the inhibition of the heme biosynthesis. So this is the different metabolites or the metabolic pathways that were altered after treating the MCF-7 cancer cell lines with the crude IL-GFE. At the end, we can conclude that the process condition of the MAE, which is the microwave assist extraction, were optimized, and the extraction yield of the 66% was achieved, and the IC50 of the crude extracts of 4.75 microgram per milliliter for MCF-7 and 10.56 microgram per milliliter for HT-29 cells were achieved. Also, the crude IL-GFE showed better performance in reducing the IC50 of both cancer cell lines compared with the purified fractions. Therefore, we have continued our experiment with the crude extracts IL-GFE. The next is the cytokinetic study showed that the crude IL-GFE had selectively inhibited the in vitro growth of MCF-7 and HT-29 by reducing the number of cell generations and increasing the doubling time with comparable results to Taxol as a positive control, which is used as commercially as a chemotherapeutic drug. For the IL-GFE crude extracts have reduced the in vivo zebrafish survival of zebrafish larvae at a relatively high dose of 250 microgram per milliliter, while no significant changes on morphology of the treated zebrafish have recorded. Next is that the crude IL-GFE has arrested the cell cycle at the G0, G1 phase and induced both mechanisms, apoptosis and necrosis, in MCF-7 and HT-29 cancer cell lines. The pathway analysis of the metabolomic profile showed an alteration of many metabolic pathways that contribute to energy metabolism and the growth of cancer cells. At the end, consequently, based on the results of the antiproliferative effect of the crude IL-GFE on cancer cells, it is strongly believed that the Graviola fruit will be a great contribution to cancer treatment in general, particularly for breast and colon cancer patients. For the future work, we recommend in vivo studies should be tested on xenograft mouse model to investigate the effectiveness of the crude extracts and the isolated fractions. Further investigation to illustrate the mechanism of underlying the therapeutic effect of the fruit extract on cancer cells by employing omic technologies and microarray analysis. More studies are needed to investigate the neurotoxicity effect of graviola by quantifying the neurotoxic molecules and determine the toxicity level to humans. And clinical studies are required to verify and validate the plant extract safety to be adopted as a therapeutic anti-cancer agent. The output of this research work is summarized here. We have published 3 papers. We have journal articles and conference papers, and different other papers are under review. One paper about optimization, another one is about the antiproliferative activity of the crude extract on HT-29, and the other paper is about the acute toxicity of the crude extracts on zebrafish developing embryos, and the other paper is about the evaluation of the metabolomic profile of breast MCF-7 in response to the crude extract treatments. This is the prototype of published papers. One published in the Journal of Ethnopharmacology, and the other one is EMGM International Medical Journal Malaysia, and this is published in the Proceeding of the ICPIOE Malaysia. This is some selected references. Thank you for your listening. Wassalamu alaikum warahmatullahi wabarakatuh.